Hi, guys. Good morning. Happy Monday. I hope you guys had a great weekend. I hope you got some rest and you're geared up and ready to go for this week. So, this week's going to be really cool. We're going to be investigating protein synthesis, and I have found a fabulous virtual lab or a simulation that's going to help us visualize this process because we can't see these things happen in the body. So, that's what's to come for this week. I'll go through your agenda with you guys in just a minute, but let's first of all take a look at what our essential question is. So, just to back it up, so we are still in segment 5. So, we're talking about structure and function and growth. And our essential question is, how does DNA direct the making of a protein? So, how does DNA direct the making of a protein? So today we're going to be using the knowledge that we've gained so far, all of the knowledge that we've gained on DNA and the structure and the function of DNA, the different molecules of RNA. We're going to put all of those together and we're going to synthesize a protein. Alrighty? So our learning target for today, so there are 2 learning targets. So our first learning target is to use a model to explain how the genetic information in the DNA molecule is transcribed into mRNA. Our second learning target, explain how mRNA is translated into a specific sequence of amino acids in a protein molecule. So these 2 learning targets are very heavy with vocabulary. There's a lot of vocabulary embedded within those. So we see transcribed, we see DNA, we see mRNA, okay? So if we take a look here at our vocabulary list, so remember we made some vocab organizers. So a while ago, I think maybe a week or so ago, I had you guys work on Quizlet and you guys made some vocab organizers with these terms. So wouldn't it be a bad idea to actually go back, since you guys are on the Chromebooks for this period of distance learning, pull this electronic organizer up, review these terms. It will assist you as you work through this virtual lab if you get stuck with any of the vocabulary. Because I had you guys add some visuals to that. So if you want to go ahead and go back and see what I'm talking about, so this is what I'm referring to. So you guys made a visual organizer. Here were your terms. And you guys went in DocHub, and our first one, for example, was protein. So if you guys have access to this in your drive, or go back to Google Classroom and pull this up, you'll see a nice image of what we're trying to do. So here we're making a protein today. All right, so the amino acids are those small little circles. And when you get a string of those small little circles, all in a chain, a polypeptide chain, that's what's going to fold and make a protein. All right. Also, some other really good visuals here that are going to help us out, and we'll see this in our interactive today. But remember the RNA molecule tRNA, so transfer RNA. So remember, this is what tRNA looks like. It's this funny kind of cross shape, all right? So this is going to be really important to help us visualize these different molecules as we see them played out in our simulation today. Okay, so if you have those out, great. They're going to be helpful for you. Okay, so our agenda. So what are we going to do today? So speaking of vocab, I want us to do a really quick vocab review from the notes that we took on Thursday. So on Thursday, we finalized those notes on transcription and translation. And you guys were to process those. If you haven't processed those yet, go ahead and take some time today and work on processing those. Add in your annotations, make your questions and notes on the side, summarize the best you can, okay? And remember that Flipgrid post for that, you'll see on Google Classroom, that's gonna be due Wednesday. So we'll vocab review. I have a really nice animation, really quick 2-minute animation of the process of protein synthesis actually in real time. So going from DNA to RNA to protein, we're going to see that actually played out before we go ahead and make our own molecule, okay? And just to back up real quick, here on our vocab, our third bullet point, protein synthesis. So this word synthesis, just remember synthesis or to synthesize, that means to make. Okay, so making a protein. So when we say protein synthesis, we're thinking we're making a protein. So we could actually reword this essential question. We could say, how does DNA direct the synthesis of a protein? Okay, how does DNA direct protein synthesis? All right, so making means synthesizing. Alrighty, and then item number 4, and again, I have the links on our agenda here like always, but remember when you go to do your work, you're going to go to Google Classroom to access those documents, type on them electronically, and then submit them through Google Classroom. Alrighty, so I have item 3, process notes. If you haven't done those yet, please be sure to do those before you interact with the virtual lab. And then item A and B under 4, those are your worksheets, okay? So under students will need, so you'll need to grab a pen or pencil, highlighter, whatever you like, pen or pencil. You'll need some paper, notebook paper, or even just a white sheet of paper is fine. Okay, I'm gonna have you guys do a notice and wonder chart on our animation, so you'll need that to reference. The codon wheel I actually have on your table. So just know that you'll be using a codon wheel today. So we'll be getting good practice with using the codon wheel. And then again, those worksheets are A and B. Okay. Alrighty, so the central dogma of biology. So reflect on Thursday. So you guys took some notes. We talked about transcription, we talked about translation. So how is information passed? So pause this video, look in your notes, and see if you can figure out what you're going to place in those blanks. And there's arrows here. So you're directing the flow of something. Alrighty? And absolutely. So how is information passed? So we're thinking genetic information. So when we hear the central dogma, we're thinking genetics. We're thinking it starts with DNA. How does genetic information flow? It goes from DNA, RNA, protein. So DNA provides the instructions for how we're built. That message is transcribed into an mRNA. That's the code. And then the code is translated into a protein, okay? And then protein, what is this protein? What do proteins do? So proteins just do something. They have various functions in the body, remember? So you go through the process of protein synthesis, your protein shows up and it does something, carries out some function in your cell. It codes for a trait. Okay, proteins can code for traits. So genes code for traits, right? That are expressed through protein synthesis. So just think of it that way. So we all have eyes. We have different color eyes. We all have hair. We have different color hair. So the color of our eyes and the color of our hair, okay, or how tall we are, right? Or if we have freckles, that's held in the DNA that's in every single cell of our body, remember. So through the process of protein synthesis, that's how traits are expressed, okay? So our genetics, our height, if we have freckles or not, the color of our hair, that's all held within the DNA in the genes, right? Remember genes are segments of DNA that are in the nucleus of every cell. Okay, so 3 more vocab terms. So you'll find these in your notes from Thursday. So when we're going from DNA to DNA, so if we're making a copy of DNA, okay, this arrow means we're just going from DNA to DNA. What's the word for that? So pause it if you want to think and reflect back to Thursday, look through your notes. Replication, absolutely. So if we're replicating a DNA molecule, or we're DNA synthesis is the same as replication. You're making another copy of DNA. You're making DNA from DNA. Okay, so DNA copies itself. That's called replication. What about if you're going from DNA to RNA? Right, transcription. So be careful with transcription and translation. They can be really easy to get mixed up. So transcription is taking the DNA message and embedding it into an RNA code. Okay. What about now we take this RNA molecule and we make a protein? Protein synthesis is called translation. Okay. So you're translating this message into a protein. Alrighty, great. I think the next slide is going to be our Notice and Wonder chart. So if you guys can go ahead and get a sheet of paper out, just a lined piece of paper or a blank white sheet, and just make a T-chart like we always do. And we'll be looking at an animation here going from DNA to RNA to protein in real time. So it's really cool. It's very short. So pause the video and set that up.
The DNA double helix contains two linear sequences of the letters A, C, G, and T, which carry coded instructions. Transcription of DNA begins with a bundle of factors assembling at the start of a gene to read off the information that will be needed to make a protein. The blue molecule is unzipping the double helix and copying one of the two strands. The yellow chain snaking out at the top is a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA letter by letter to copy the gene. At this point, the RNA needs to be edited before it can be translated into a protein. This editing process is called splicing, which involves removing the green non-coding regions called introns, leaving only the yellow protein-coding exons. Splicing begins with assembly of factors at the intron-exon borders, which act as beacons to guide small proteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released, and the spliceosome disassembles. This process is repeated.
So remember, guys, you're writing what do you notice and what do you wonder. So make sure you're writing down some things that you notice and some things that you're wondering about.
Repeated for every intron in the RNA. Numerous spliceosomes remove all introns so that the edited RNA contains only exons, which are the complete instructions for the protein. Again, this is happening in real time. When the RNA copy is complete, it snakes out into the outer part of the cell. Then all the components of a molecular factory called a ribosome lock together around the RNA. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. Inside the ribosome, the RNA is pulled through like a tape. There are different transfer molecules for each of the 20 amino acids shown as small red tips. The code for each amino acid is read off the RNA 3 letters at a time and matched to 3 corresponding letters on the transfer molecules. The amino acid is added to the growing protein chain, and after a few seconds, the protein starts to emerge from the ribosome. Ribosomes can make many proteins. It just depends what genetic message you feed into the RNA.
All right, great. So I hope you jotted down some notes, some notice. What did you notice? What did you wonder? If you need to go back and revisit that, you'll find that link on Google Classroom, okay? Again, we're going to now go into our steps of the virtual lab. And also really quickly, speaking of, I mentioned I wanted you guys to finish these notes. So I also linked the notes here, guys, from last week. I just added a couple slides. I made a copy and I added a couple slides. So you'll also have access to these if you're viewing this to look at the links, okay, so you have this in Google Classroom, so you don't have to go searching too far. But just to refresh your memory, guys, so you'll see your post-lab questions that you'll do at the end of the exercise, and then here is the link to the interactive. So these are what you're going to open up and type in your Google Doc when you're finished with the lab. Okay, great. One more quick thing. So please don't forget to do your Notice and Wonder. Review the animation if you need to again, because you're going to be using this Notice and Wonder chart to contribute to a discussion. Alrighty, so I will open up that post later on this morning, but make sure you have something on your Notice and Wonder chart, and you'll see a Google post on this. Okay, so we have some steps. So number one, go to Google Classroom and access the link. You have access to the table, the code on wheel, and the journal questions as I just showed you guys. Number two, read the background information, objectives, and procedure. Number three, follow the procedure to complete the lab. Number four, complete your table. You're going to be entering in some data, and I'll show you guys, I'll demo how you're going to go ahead and set up that table. Number five, complete the journal questions. Those are post-lab. These post-lab journal questions are the same thing. So complete the journal questions. Those are due Friday. Okay, those are due Friday. And then question number 9 of your post-lab questions, you guys are going to do a Flipgrid post. All right, so you're going to go ahead and verbalize and reflect your knowledge on that question. So that's coming up later this week. So if you click on the link to the animation, to the lab, you have to go ahead and enable Adobe on your Chromes, and you will be presented with a screen like this. So you see here you have some text, some paragraphs here. So this is your background information and your procedure. So your objectives start here and your procedures right under that. Now, what I went ahead and did is I made a document for you guys. It's easier to read on here, but I do like that the Lab Interactive has the instructions right here so you can follow along and just do exactly what the procedure says, okay? Your background reading starts up here. So I made a separate doc that you'll also see on your Google Classroom. I made a separate doc, and the same information that's on the left-hand side of that tab is all right here. Okay, so this is all review information that we've been discussing for quite some time now. I'm going to jump down, and you guys can pause this at any time. Remember, you're going to read the background information and process those notes before you engage in this interactive. So pause the video if you need to go ahead and do that, or wait till it's done and I give some instructions on how to complete the lab, and then you can go back. Okay, so you have access to this screencast, so you can stop it and watch it as many times as you want. So again, just restating our learning targets, explain how genetic information in DNA is transcribed into mRNA, and explain how mRNA is translated into a sequence of amino acids in a protein. So our procedure here, number 1, says observe the unwoven DNA molecule. One of the DNA strands is exposed, showing a sequence of nitrogen bases. All right. I keep hitting that one. Okay. So you guys read the background info on your own. And I'm just going to pull up the procedure so we can have it at a glance, okay? So what we're looking at here, we're looking at, what do you guys think? Yep, DNA. Double helix, you've got your bases in the middle here, you've got your sugar phosphate backbone, that green ribbon-like structure on the sides. So this DNA is unwoven, so it's unwound. There's a replication fork here. And you've got these bases, these nitrogen bases in DNA. And we know that because there's T's. So we have some corresponding bases over here. These are RNA bases. And we know that why? Well, we don't see a T. There's just a U. Remember, uracil is used in RNA instead of thymine. So what you guys need to do is use your base pairing rules for RNA and complementary base pair your RNA to your DNA. So what matches with T? Oh, that's loud. Take our A. We have an A here. U goes with A. Go ahead and synthesize this molecule using your knowledge of RNA base pair rules. Back with you in just a sec. All right, guys, so I'm almost done synthesizing my molecule. So pay close attention when you bring in this last base. You're going to see the protein synthesis carried out. It's coming out of the nucleus, finds the ribosome. Those tRNA molecules come in and drop off the amino acid, and it moves right along. This is your mRNA.
Now, figure out the code.
All right, and if you hit show labels, you'll be able to see that these molecules are all labeled. So your RNA is here, your ribosome, and what you're given is a sequence of amino acids. So once you get here, your task is to go back to your document that you guys have on Google Classroom, and you have this table, okay? So everybody is going to start with amino acid 1. However, the sequence is going to be different for all students. So this simulation is going to give you a different amino acid sequence every time you go ahead and complete this simulation. And I'm hoping you'll do it a few times. So we know that the mRNA code is AUG because we can see AUG right here, and that's corresponding to amino acid 1. So in my table, I'm putting amino acid 1. My mRNA code was AUG, and your task is to now go back and determine, well, what was the DNA code that it started with?